Okay. Ladies and gentlemen, good afternoon. Let me welcome you at today's MENDEL lecture, which will be given by Professor Xie from Harvard University. Sunney graduated from Beijing University in China, and then he moved for Ph.D. to University of California. After that, he took 3 years postdoc at the Chicago University. From there, he moved to Northwest Pacific National Laboratory in the States, where he worked as a senior scientist for a number of years before taking a position of professor at Harvard at the Department of Chemistry and Chemical Biology. And now he's Mallinckrodt Professor of Chemistry at this university. 3 years ago, he took another position. So he has now two positions, one at Harvard and second at the Peking University at the research center. And as we discussed over the lunch, he is considering increasing his activity in Beijing. He is known for his work in single molecule techniques. He is one of the founders of the field, and he was interested in applying these techniques, single molecule techniques at the room temperature to study various biological processes like activity of enzymes. He was first to show that the enzymes have certain periods of time when they are resting, they are not active, and then other times when they are active. So that was one of very seminal work in the field of enzymology. He also applied single molecule techniques in the field of genomics, which will be focus of his talk today. He received a number of awards. I cannot name them all, 11 awards altogether. Last 10 years, every year at least one award, which means he's widely recognized in scientific community for his work. So we look forward for your talk. Sunny, the floor is yours.
Well, thank you, Jerry. I would like to thank Jerry and other organizers give me the opportunity to be here today. I just had a tour of this amazing place. I felt the epiphany at this birthplace of modern genetics. A man who had so much influence to our field, yet he wasn't quite recognized or awarded before his death. You see this time axis from MENDEL to the determination of human genome. Really the starting place was here. So Jerry mentioned that I have been working on single molecule biology. I started my independent career at a very exciting time when imaging of single molecule at room temperature was contemplated. This was probably the work that got me a job at Harvard. Former postdoc Peter Lu was able to monitor the enzymatic turnovers of single enzyme molecule of cholesterol oxidase, which catalyzed the oxidation of cholesterol by oxygen. The enzyme has a flamin, which is fluorescent in its oxidized form, but not fluorescent in its reduced form. So when we view the emission of an immobilized enzyme molecule, the emission is turned on and off, on and off. Each on-off cycle corresponds to enzymatic cycle. So we're viewing the enzymatic reactions of a single enzyme molecule in real time. The signal looks just like the single ion channel signal recorded by the patch clamp technique. That was the first single molecule experiment in biology. But we were monitoring chemical reactions of a single enzyme. Later on, at least two groups use individual DNA polymerase molecules, build single molecule sequencers to sequence DNA. But many fundamental research have come out from single molecule research. So more than a year ago, I wrote a review article at the occasion of 100-year anniversary of the Michaelis Menten equation. I started showing these photos when one of my students told me that he thought Michaelis Menten was one person. Michaelis was a famous entomologist in Germany. Menten was a biochemistry student. She went to Germany to study with Michaelis. After this celebrated equation, she has made many other contributions to science and eventually worked at the Pittsburgh Medical School in US and then became one of the first women professors in Canada. So when we analyze the single enzyme traces, we found that an enzyme like us works hard for a while, then it slows down. It doesn't have constant activity. Nevertheless, the Michaelis Menten equation still holds at a single molecule level. This bracket indicates the average of this stochastic enzymatic cycle time. This time trace, unlike the conventional experiment, is not reproducible, even though the statistical properties are reproducible. So by analyzing these time traces, we learn a great deal about how enzymes work at the molecular level. So this fluctuation of the K-cat is due to the conformational fluctuations that are otherwise hidden in the ensemble averaged measurements. Now, there are many reasons one would want to do single molecule experiments in biological systems. But I think a major reason is DNA. They exist as single molecules in individual cells. Because of that, gene expression must occur stochastically, just like the single enzyme trace I showed you. So in 2006, my research group at Harvard reported this real-time movie of protein production. Each yellow flash you see here is due to one copy of yellow fluorescent protein molecule spontaneously generated by gene expression. By analyzing these time traces, we were able to quantitatively describe the central dogma of molecular biology, the making of messenger RNA based on the genetic information in DNA in the transcription process and the making of protein in this translation process. Now, this movie was recorded under the repressed condition. The genes are not induced, so we were watching the leakage of gene expression. Not surprisingly, the gene expression was stochastic. Only you have one transcript made when the repressor molecule dissociates from the specific sequence on DNA, and then a few copies of protein. Now, under the partially induced condition, you see these bacterial cells have two different phenotypes, fluorescent or not. We found that this cell switched from one phenotype to the other, and we proved that this was due to the stochastic event of a single molecule. Like repressor, dissociate from one operator followed by the complete dissociation from another operator, the specific sequence, that result in this life-changing decision of the cell. So experiments like this indicated the importance of single molecule studies. Many phenotypic changes in cells come from this stochastic single molecule events. So what about under induced condition? That is to say the gene is fully induced. We and many other people have found that the production of messenger RNA and the production of the protein are still bursty. But the mechanism was not known. This was a ubiquitous phenomena. Every induced gene in a bacteria, every bacteria we studied, And this is even true for mammalian cells, have this bursty gene expression. The mRNA production is bursty. Now, it took us quite a few years to understand the molecular mechanism, and I'm going to describe to you this mechanism in the next 10 minutes or so, just to show you the direct observation of mRNA production, mRNA bursts in this real-time experiment done by Ido Golding's group, and many other groups have seen the bursty protein production in mammalian cells as well. Okay, but this was a very difficult puzzle to solve. We had to wait until the collaboration with a very bright colleague of mine, Xiaoliang Zhuang. We published a paper a few years ago using her super resolution microscope. We studied this histone-like protein in bacteria called HNS. We found that HNS have two clusters inside the E. coli cell. In 1960s, electron micrographs has shown that the circular DNA in E. coli, is organized in these DNA loops. So there are about 400 HNS binding sites that form 400 DNA loops, each about 10,000 base pairs, containing at least one operon. Now, the biochemist Jim Wang, who used to be at Harvard, made a discovery several decades ago of topoisomerase. He pointed out if two ends of the DNA are fixed, then when the RNA polymerase generates a messenger RNA transcript in the backward direction, you would have negative supercoiling. This supercoiling could be devastating. If it's not removed, the RNA could hybridize to the parent DNA. Jim discovered the enzyme that quickly removes the negative supercoiling, topo1. That is a very fast enzyme. In the forward direction, there's positive supercoiling. Several years later, gyrus was discovered. It is ATP-dependent enzyme removing the positive supercoiling. So these two enzymes are needed in order for transcription to take place. Let me quickly remind you that the transcription process starts with initiation, the binding of RNA polymerase on the promoter, the specific sequence on DNA, followed by elongation, the linear movement of RNA along the DNA. And then when the transcript is made, it ends by the termination, the dissociation of RNA polymerase and the messenger RNA from the template. Shasha Chung and Chongyi Chen, two former graduate students in my lab, developed an assay that allows us to make real-time observation of RNA polymerase making transcripts in vitro. They found this dye molecule that binds to RNA preferentially over DNA. So if we monitor at this emission wavelengths, you will see the emission from RNA. So in this field of view, they observe these spots. We have DNA duplex tethered to the glass surface and flowing RNA polymerase. Focus on a particular spot, you see the intensity linearly ramping up because of the making of the transcript. The longer the transcript, the brighter the fluorescence. It is followed by the abrupt dissociation, that is termination. So from this, we could measure the elongation rate. Now, in this experiment, the DNA double helix is tethered to the glass surface using the single linkage of biotin and strep-avidin. So the double helix could rotate with respect to the surface. If we use this multiple linkage, the rotation is stopped, then the positive supercoiling would build up. Unlike this single linkage, the positive supercoiling cannot build up. So the elongation rate is slowed down. Then we flow in the gyrus, the positive supercoiling is removed, the elongation rate is recovered to the value of a single linkage. So this titration allows us to determine the KD of the gyrus binding to this DNA fragment. The concentration of gyrus at half of the elongation rate of the saturation, about 100 nanomolar, is the KD of the gyrus binding. This is similar to the Michaelis Menten equation. So what we have learned is positive supercoiling slows down elongation, but it doesn't stop elongation. What about initiation? What is the effect of supercoiling on initiation? How would they measure that? Xiaoliang and Cheung Yi built this second assay using circular DNA multiple linkage. So in this field of view, we count the number of bright spots. Now, if we add no topo 1 nor gyrus, we have positive supercoiling, negative supercoiling. When the transcript is made, then this positive and negative supercoiling cancels, so the number of spots would be constant. Doesn't change with time, it's a steady state value. And then we flow in topo 1, which quickly removes the negative supercoiling. And the positive supercoiling would build up and cannot be removed. So after one transcript, another one would give more supercoiling. So this build-up supercoiling results in the reduction of the number of bright spots in the field of view. This decays down almost to 0, which means supercoiling can stop the initiation. As a control experiment, we flow in gyrus, positive supercoiling is removed, and then this bright spots will reduce to the initial value. Now from this rise, we were able to determine the bimolecular rate constant for the gyrus binding to the DNA loop. So to put everything together, based on the fact that the transcriptional initiation is stopped and restarted by the gyrus binding, we provided this mechanism. Basically, the gene has two different states. State 1 on, state 2 is off. Now, people knew there were two states. They just didn't know what was the molecular mechanism of this off state. We now know it is due to the dissociation of the gyrus from the DNA loop and the rebinding, the stochastic rebinding of that, resulting in the bursty mRNA production. Now, the number of gyrus inside the cell is about 400. So each loop, on average, gets one gyrus. If there's only one circular DNA, you wouldn't need that many gyrus. So this was an in vitro experiment. Is it really what's going on inside the cell? We use RNA fish experiment to measure the transcripts, number of transcripts inside the cell in live cells. And we found, from that experiment, we could determine the branching ratio, this alpha and beta. Beta is the bimolecular rate constant, which is dependent on the concentration of gyrus. So if we overexpress gyrus, then beta is increased. If we inhibit gyrus, the beta is reduced. So in this wild type experiment, we add gyrus inhibitor, then this branching ratio changes. And this is global, all the genes changes. So this is really a global behavior. It is a single molecule behavior because it only occurs at one DNA locus. Now, what I don't have here is that when we overexpress gyrus, all the red ones are higher than the blue ones. So this is in vivo proof of this mechanism. So let me quickly summarize. Transcriptional bursting in bacteria is universal for all highly expressed genes and is associated with DNA loops that define topological barriers for transcription. DNA positive supercoiling generated by transcription in the absence of gyrus slows down both transcription initiation and elongation, but eventually stops initiation. Transcriptional bursting in bacteria is primarily caused by accumulation of DNA positive supercoiling upon stochastic dissociation of a gyrus from a DNA segment and rebinding of a gyrus to the chromosomal DNA loop. So this is an example of single molecule enzymology, allowing us to solve in vivo biochemistry mechanisms. It is also an example that the mechanical property of DNA supercoiling here really changes the biochemistry. When you twist the DNA, the binding affinity of the RNA polymerase on promoter is affected such that it doesn't bind. This is a paper was published a few months ago, which is so that I give you an example of single molecule enzymology. Now I would like to tell you our latest single molecule experiment. I mentioned to you that two groups built single molecule sequencers. There were two commercial sequencers. One of them was discontinued and another one still working. We all hoped that single molecule enzymology would help society by DNA sequencing. But it is fair to say that the single molecule sequencers could not compete with these monster sequencers, bulk sequencers, on their throughput and accuracy. So this is after 2003, human genome. So what happened after we had a human genome? A lot happened. Starting from 2007, the cost and the time it needed to sequence a human genome has been reducing faster than the exponential decay, the Moore's law seen in the semiconductor industry. Because of this, we're in a position to have personalized genomics and now precision medicine. Why does your DNA make you you? How do you prevent diseases or conquer diseases? So I have a personal example. My daughters are identical twins. Of course, they are very similar, but they're not the same. They have not only different footprints, but also different characteristics. I used to think that the fact they have different characteristics has something to do with stochasticity of gene expression. It turned out that a few years ago, there was a report stating that identical twins do not have identical genomes. People used to think identical twins have identical genomes. Why? Because our genomes are dynamical, changing with time. We did not have the tools to study them. I mentioned stochastic events in biology. It can result in, can have important biological consequences. Right? Mutation is an example. So, in a human cell, there are 46 individual DNA molecules, 23 from each parent, 46 chromosome molecules. They all have different sequences. So what are the stochastic changes taking place on this DNA? Well, two major ones. The first is copy number variation. This was a phenomenon that was really discovered only 8 years ago because of the genomic studies. Although people knew from chromosome counting, it must exist, but it was the genome-wide study was only carried out a few years ago. So what is that? So normally the copy number of a particular gene is 2, because you get each one from you get one from each parent. But in certain cells, cancer cells in particular, the copy number is not 2, it's 1 or 3 or 4. Okay, so that's the copy number variation. Single nucleotide variation are the point mutations. At the end of 2013, Nature Methods magazine made the single-cell genomics as the method of the year. In the past few years, there have been tremendous development. Most of the work have been focusing on single-cell transcriptome analysis. We developed a method to do single-cell whole genome amplification. So we want to determine the genome of a single cell. You give me a human cell, I want to be able to report to you pretty much the entire genome, the copy number variation, and the point mutations. Now at this time, there isn't a method that would allow us to, a single molecule method that would allow us to sequence the 46 individual DNA molecules from beginning to the end. If you break them to different fragments, you wouldn't be able to capture all of them. So we still have to rely on whole genome amplification, followed by next generation sequencing. So the amplification has to be even. The first attempt was made by the PCR method. So PCR was a Nobel Prize. It's been 30 years of PCR. It has done so much for biology and medicine. It has single copy sensitivity. If there's only one copy of DNA, a gene at the crime scene, you will be able to amplify the signal to the level you can detect a good signal. However, if you want to have a whole genome amplification, there's strong sequence-dependent bias. 2 genes, if they differ a little bit in the amplification yield, because PCR is exponential amplification, 1 becomes 2, 2 to 4. At the end, some genes are amplified 1 million times higher than other genes. At the time, sequencing is still expensive. You wouldn't want to do that. It has at the beginning it had only 6%. Usually it's 6% coverage. That is not the whole genome. Okay. 14 years ago, Roger Laskin developed another method, multiple displacement amplification, which significantly improved the coverage, and it has been widely used in the past decade. But it's still an exponential amplification. It still has a sequence-dependent bias. At the end of 2012, my research group reported multiple annealing and looping-based amplification cycles, MALBAC, like the mine MALBAC. This was the work of Cheung Kong Visiting, former postdoc who's now a professor at Baylor's College of Medicine, and Shijia Lu, a former graduate student who has come back to China and co-started Econ Genomics with me. This was my disclosure of potential conflict of interest. So this method has a more even amplification, higher coverage, because it is primarily a linear amplification. So we use this DNA hairpins to protect the amplification products such that they don't get amplified, and only amplify the genomic DNA after 5 to 10 cycles, we use PCR to amplify this protected products to the level of DNA needed for genome sequencing. So this was data at the time. It's a single cell from a cancer cell line, chromosome 1 to 23 with alternating colors, blue and red. You see the copy number here is not 2, but 3 or 4 and 1. Each cell is different. Look at these 3 box regions, they're different. The bulk measurement only gave the average of these cells. So here's the MDA run. You see this noise, again, due to over-amplification in certain regions and lack of amplification in other regions. But I want to emphasize this noise along the genome is not reproducible. The sequence dependence is not reproducible, whereas our noise here is reproducible. So we could normalize this along the genome. And I'll show you the normalized data. It is quantized. It's really digital counting of the copy number. What about SMV, the point mutation? This cell had 1 point mutation, and we knew it was not due to false positive. This cell has 35 SMVs, point mutations, and they were all confirmed to be correct by Sackler sequencing. So we don't have false positive here. We do have false negative. That is to say the detectability is 70%. If the SNP is there, there's 30% chance we don't detect it. But once we detect it, it would not be wrong. We're still trying to improve the false negative. All right, this cell was picked from this bulk that was derived from this 1 ancestor cells after 20 cell divisions. So we saw 35 of them. The detectability is 70%, so 35 divided by 0.7, it was 50. So after 20 divisions, we have 50. Each time the cell is divided, the chromosome is open, subject for DNA damage. And if the DNA repair is not successful, then you would have point mutation. So we were able to measure directly the whole genome point mutation rate to be 2.5 SMV per cell division. And this number is consistent with the previous report derived from indirect measurements, but we were able to measure this directly. At this rate, it will take a long time for a cancer to develop. If I have time, I can come back to this point. So simultaneously with the Malbec paper, we had a paper studying sequencing individual sperm cells. So at the time, my Harvard group was learning genomics. It was very helpful to have a new set of colleagues at Peking University, where I've been spending a lot of time. My colleagues, Cheng Li, a bioinformaticist, and Futuo Tan, who pioneered single-cell transcriptome analysis. I recruited him to Peking University. My students and postdocs went to BIOPIC. We were able to sequence individual sperm cells. Each sperm cell has a different sequence. Everyone has 50% of the genome from the father of the donor and 50% from mother of the donor. But the crossing points are all different. The green is the paternal DNA, the red is maternal DNA. And this crossover point, as shown here, the crossover frequency is 1.2 per chromosome. people knew that, but it was just nice to see this directly. And I should say, Steve Quick's group also reported this a few months before ours using a different method. So we were also able to phase the genome of the donor. So what do I mean by that? So if I just sequence the blood of the donor, the diploid cells, you see this SNPs, point mutations, but you wouldn't know it comes from the father's allele or mother's allele. You can imagine if two of them, C and G matters, if they're nearby, if they are on the same protein or they're on the same allele or on different alleles, they would have different consequences. So one of the reviewers said to us that, why? We were able to read out by sequencing this sperm cells by simply lining up these sequences, realizing that the switching between red and green only occurs infrequently. So we can phase the genome knowing the haplotype of the donor. So one of the reviewers said to us, why do you have to do this? Why don't you just sequence the parents of the donor? We did that in order to confirm this works. As I will show you in a moment, this turned out to be extremely useful, but we nor the reviewer knew. So this donor was an American professor with Chinese origin. His graduate student was more interested than him in finding out whether he was normal. Now, this sperm cell is not normal. It's missing chromosome 19. And this one is not normal because it has 2 copies of chromosome 6. Luckily, this guy was still normal. Any man would have 5% of the sperms that are abnormal like this because of chromosome segregation errors. It's called aneuploidy. Sperm cells like this would result in birth difficulty or Down syndromes or other genetic disorder. But this 5% does not change with the age of a male. That's not to say the genome of sperms the sperm cells do not degrade with time. Point mutations are accumulated. We cannot measure the point mutations of haploid cells. The result I showed you earlier was on diploid cells. We are working very hard on this. We want to be able to measure the point mutations in sperm cells because it has been implicated that these point mutations are associated with some neurodegenerative diseases, such as autism, because autism has been linked to father's ages. Okay, in clear contrast, aneuploidy, the chromosome abnormality for female, rises drastically with the age of a female, which results in this decrease of live birth rate as a function of female's age. So IVF, in vitro fertilization, has been 36 years. This was a photo of Edwards, one of the pioneers of IVF, taken at the occasion of 35th birthday of Louis Brown, the first test tube baby, and her child, her mom. Edward was not recognized. You can imagine the difficulty he had. So many things he had to fight. By no means only scientific, right? He was only awarded Nobel Prize 2 or 3 years before his death, and was knighted. 7 million babies around the world have been born this way. Pre-implantation genomic diagnosis has started. That is to screen the fertilized egg to select one that is viable for implantation, avoiding the chromosome abnormality discussed earlier. So for many of you, this is probably just a refreshment. Human oocyte has 4 double helix, 2 from the father side, 2 from the mother side, and the crossover takes place, and upon fertilization with a sperm. In the normal case, there's a fertilized egg that has a female pronucleus, that's the DNA from the oocyte, and male pronucleus, and the DNA from the sperm. And there's 2 smaller cells, polar body 1 and polar body 2. They are dispensable. They're not needed in the IVF process. They would just be degraded. So in the case of a healthy baby, we have this situation. In the case of a chromosome abnormality, this copy number is wrong, and there will be a birth difficulty or genetic disorder such as Down syndrome. There's another situation that is the point mutation, the devastating point, Mendelian disease, single gene disease. There are 7,000 single gene diseases, Mendelian diseases. So this point mutation, there are 7,000, but 400 of them more common. So they could be passed to the next generation. It would not pass to the generation if this point mutation goes to the polar bodies. But it would, in this case, if it's in the fertilized egg, it would. And the baby would have the dominant disease allele. The word dominant was first coined here. And recessive, right? So with Malbec, what we would like to do is to avoid these 2 situations. People have been trying to use DNA microarray or FISH or PCR to avoid this chromosome abnormality. But so far, to the best of our knowledge, nobody could do both. This is hard. You must have the high accuracy, right? You need the accuracy for copy number variation to do this. You need the accuracy of calling SNPs. We want to avoid false positive and false negative. Cannot make any mistakes. This work is done in collaboration with Dr. Jie Xiao, the head of PKU Third Hospital, her assistant, Fu Chou Tang, my colleague, her student, my graduate student working on bioinformatics. It was followed by a group at BIOPIC. Shijia also contributed. When I went to Dr. Qiao for help, as soon as she heard about the experiment, she was so excited and convinced me that studying women is a lot more interesting than studying men. So that's how we started. And it took us only a year to publish this paper that provided proof of principle for this experiment. So if the problem comes from the mother, we would do this. This is 2 polar bodies, the fertilized egg. Using laser to drill a hole, the pipette comes in, sucking out the 2 polar bodies, and we would sequence these 2 polar bodies because they're dispensable, and we would use this information to deduce what's in the fertilized egg is viable for implantation. The female pronucleus is viable for implantation or not. If the problem comes from the father, then we would take a few cells at this blastocyst stage in day 5 of the embryo. Of course, if the problem comes from mother, we could do the same, but we have less time here, only a day or 2. So in the 2013 paper, we show the sequence of second polar body, which is haploid like sperm. So we see the crossovers between the paternal and maternal DNA, the green and red. You see the frequency of the crossover for the second polar body here is almost, it's 1.9, it's almost 2. You know, again, people knew that, but nobody could see. It's the first time they see the human sequence. And each oocyte is different. And this is why you really need single cell. You need single cell because the sample is so precious. You only have one cell to work with. Okay, so the first polar body is diploid. That is the left of this. This is one oocyte, 23 chromosomes. The left one is the first polar body. Yellow means the combination of red and green. The red one, the right one is the first polar body. It's haploids, either green or red. So we sequence 2 polar bodies, and we could deduce what's in the female pronucleus. Under the consent of the donor, we take the female pronucleus out and sequence them. Let's compare the prediction and the experimental verification. They're identical, which means this oocyte is viable for implantation. Not all of them are. Let me show you another one in a different way. Chromosome 1 to 23 with alternating red, blue colors. See, the first polar body is supposed to be 2, but it's higher here, lower here. Second polar body is supposed to be 1, is lower here. With this 2, we could deduce what's in the female pronucleus. I want to say this is the normalized sample. You see, it's really digitized. There's essentially no noise. That's the prediction, and that's the experimental validation. Again, it's flat. Okay, of course, this prediction is based on the fact there are 4 double helix, and that number 4 is conserved. If you sum the first and second and female pronucleus, then number 4 should be constant along the genome. Okay, what about point mutations? If we see point mutations in the first and second polar bodies, we would be able to deduce the point mutations in the female pronucleus. So we can use, so here's where we use this the same experiment as the sperm by sequencing 2 or 3, actually at least 2, 3 will be helpful. By sequencing 3 second polar bodies, we'll be able to face the genome of the oocyte donor, just like we did for the sperm. This is useful because people don't always get to sequence their parents. Okay, so this allows us to see the point mutations of the fertilized egg, which is necessary for the Mendelian diseases. So a clinical trial has been started at Peking University Hospital by Dr. Cheung. Of course, we could do chromosome abnormality. Other people could do it with DNA microarray. But most of our patients at early stage of the trial had Mendelian diseases. So what we have completed were this 15 cases. So this is all due to single point mutation, or this disease is all due to single gene. So let me show you the first case. This man wrote to me about 2 years ago after his friend told him about our paper. He suffered from this bone cancer, all because of the point mutation at EXT2 gene that he got from his father, and his father got his from his grandfather. His wife is fine. Every year he would need a surgery. His body is full of metal, and of course he doesn't want this pass to his next generation. So he really did his homework, gave me all the references, and luckily this was well-studied gene. But what he wanted to do, of course, is avoid heterozygous mutation, right? So we need to select one, and that gene only comes from his wife, but not him. We need to select that embryo. In this cycle, so she had 18 oocytes. So you can see this chromosome abnormality. This was done with 2x sequencing depth, so relatively cheap. We only need to do 2x for each embryo and 2x for each parent and the grandfather. Using linkage analysis, we could do all the phasing, just very similar to the phasing I discussed earlier. So it's actually not that expensive. 5 of them had neither undesired point mutation nor aneuploidy. We picked number 4. I was often asked this question, why number 4? Each one is different. Because it looks good, it looks healthy. The young people here think about planning a family. Let's not get too far ahead. We're only doing this for this devastating genetic diseases. I think no matter in what country, what culture, religion, I mean, this is really necessary. So I was there when the ultrasound was done. You can imagine how happy the parents were. And in September, the baby was born. This was Dr. Chow and my colleague, Fucho. The baby was more than perfect. She was smiling at us. It didn't even cry once. And of course, we knew when we had the MU test that she was completely fine. And after her birth, everything was normal, and the disease would not be passed to the future generations. Okay, the second case was a problem from the mother's side. This actor brought awareness of this, another Mendelian disease. The symptom is no hair, no hair follicles. So the point mutation at this EDA1 gene from the wife was a problem. And their first baby also suffered from the same disease. Because of no hair follicle, so he had a high fever, and that resulted in mental retardedness. So they really worry about this. So of her 5 oocytes, these 2 green ones had neither aneuploidy nor undesired point mutation. And in November of last year, she was born, again, perfect. So we now have 3 babies born, no problem. 35 of them have been enrolled in the process too. So in summary, by avoiding aneuploidy and undesired point mutations, we're hoping we will see the increase of the successful rate for healthy babies. So if I still have 5 minutes, let me tell you another application of single-cell genomics to cancer. Cancer is a horrible disease that has still taken so many lives every year. Cancer is a genomic disease. So it is important to do genome sequencing of individual cells, but cancer cells are very heterogeneous. Cancer genomics so far has seen heterogeneity of heterogeneity. So we wanted to study circulating tumor cells, which exist in low copies, really need the single cell technology. These cells come from the primary tissue, enter into the circulating system, lead metastasis, which is responsible for 90% of cancer-related deaths. So this work was done in collaboration with Dr. Jie Wang of Beijing Cancer Hospital and her colleague, Dr. Lu, in collaboration with Professor Fan at BIOPIC, and my postdocs went to BIOPIC. Recently, we have been collaborating with Dr. Zhang in Tianjin. So the first patient was lung cancer. We sequenced 8 circulating tumor cells. When we look at the point mutation, these are the point mutation on these oncogenes, tumor suppressor genes, and there's some overlap with the primary tissue. This patient has metastasis, also some overlap with the metastatic site, but there are some differences. But I want to emphasize here is, again, you see the, for point mutation, heterogeneity of heterogeneity. It's not reproducible. And if it's not reproducible, what is the use of it for diagnosis or even monitoring? However, we made an interesting discovery. These 8 circulating tumor cells have very similar copy number variation patterns. You see these ups and downs, chromosome 1 to 23, alternating colors of blue and red. They are the same, and they are the same as the metastatic tissue. This is bulk measurement, but not the same of the primary tissue. Now this is because the primary tissue is heterogeneous. Okay, this means that only certain cells from primary tissue enter into the bloodstream. They're selected. Okay, now you can say, oh, this is all from one clone. That's why they are the same. But what we found is this: patients, different age, different sex, with similar cancer type, have similar patterns, but patients with different cancers have different patterns. Okay? So this is very interesting. This suggests that this pattern is tissue specific or cancer specific. They are the same as the metastatic tissue. It's not coming back to the circulating system from the metastatic tissue because point mutations are different. But this copy number variation is the same. This is something people didn't know. They didn't have the technique. Okay, so this suggests that when you change the gene dosage, is the copy number, the system biology of the cell changes. Then the DNA repair system will not work, and that result in point mutations later on. But these point mutations are not reproducible. But these copy numbers are reproducible. So this is actually, I want to say this is not, we can back this up by statistical analysis. It's not just visual. And now we, in collaboration with the Tianjin team, we found this, for these different cancers, these patterns are different. So the conclusion is in the evolution of cancer, copy number variation of certain chromosome regions are selected for metastasis. And this pattern is tissue or cancer specific. And this, of course, this reproducibility of cancer-specific CMVs offers potential for CTC-based cancer diagnosis. The problem is we must detect them at an early stage. There are probably 50 groups working on collection of circulating tumor cells. We did this using a commercial unit based on the surface antibody. We don't collect many of them. The goal is to make it very sensitive. If we could pick up just a few at an early stage, then this can be used as a diagnosis based on this copy number value. We can tell which kind of cancer it is without doing a surgery, without any operation. So, in summary, DNA exists as single molecules in individual cells. Because of this, gene expression is stochastic, even under induced condition. Single molecule entomology allows us to study the in vivo mechanisms of this fundamental processes, transcription and translation. Single-cell whole genome amplification is where single molecules meets genomics, meets genetics. And I hope I have convinced you that the ability to count 1, 2, 3 in a living cell is not only now possible, but it's also critically important. I'm delighted to show you here an example of a single molecule technology allow us to probe and understand a better life at the single molecule level, which I believe will have an impact to human health. I wouldn't be able to tell you these stories if I did not work with a group of very talented graduate students and postdocs. This is my current group, my small group at BIOPIC, and these are the colleagues, my new colleagues at BIOPIC. We're currently on the second floor of this building. This is a new building they're building for us. And I want to thank Jerry and other organizers for giving me the opportunity to come to this special place. I feel the epiphany here, and I thank you all for coming.
Nice science, but also the applications were beautiful. As a last point, I would like to mention that Sunney was traveling to China early this week, and he's traveling back to China tomorrow and then back to United States. So it was really remarkable that he made this trip to give a lecture here, and as an appreciation of that, we want to present a medal to you. Mendel's medal.
Oh, thank you.
To commemorate your lecture.
Thanks so much.
Thank you for joining us from the campus and from Institute of Molecular Genetics in Prague, and thank you all for a nice evening.
Thank you.